All right, buckle up, because today we're pulling back the curtain on something that truly blurs the line between science fiction and tomorrow's medicine, gene editing.
Specifically, the groundbreaking technology called CRISPR.
Yeah, CRISPR. We're going to trace its wild ride, from its surprising origins to its astonishing applications now.
And grapple with the profound ethical dilemmas it's already creating.
We are.
And we've compiled quite a stack of the most relevant sources, things like cutting-edge scientific reviews, breaking news,
Right.
to give you a really clear, structured understanding of how this technology works, the persistent challenges it faces, and the incredible, highly personalized breakthroughs happening, well, right now.
So our mission today
Is really to distill the most impactful insights so you walk away truly well-informed.
Okay, so to really understand where we are with CRISPR, we kind of have to look back, don't we? Gene therapy, the whole idea, it has a surprisingly dramatic history.
Oh, absolutely.
What did those early attempts look like?
You're right. The early days were full of both immense hope and significant turbulence. Initially, the focus was on using viruses.
Viruses.
Yeah, tweaked to be harmless, hopefully, to deliver new working genes into patients. We saw pioneering clinical trials back in the 1990s.
For things like?
Conditions like melanoma and AD-SCID, actually the very first clinical trial with therapeutic intent for AD-SCID, that was back in 1990, delivering a corrective gene into T lymphocytes.
But then, well, things didn't go smoothly. The field faced some really deeply tragic setbacks.
They did.
These moments forced a major reevaluation, right?
Oh.
Can you walk us through those pivotal incidents?
Unfortunately, yes. That early promise was quickly overshadowed by severe challenges. The most prominent, sadly, was the death of Jesse Gelsinger in 1999.
In a clinical trial.
In a trial, he suffered a fatal immune response triggered by the adenoviral vector, the virus used to deliver the gene.
Wow.
And soon after that, in another trial for SCID, several children developed leukemia.
Leukemia?
It was due to something called insertional oncogenesis. Basically, the therapeutic gene accidentally slotted itself into a bad spot in the genome.
A proto-oncogene locusus.
Exactly, a gene that, when disrupted, can trigger uncontrolled cell growth. Cancer.
So those events,
Yeah.
they must have just stopped everything.
Pretty much, it led to widespread concerns, a major pause for the entire field. Safety, informed consent, huge questions.
So with those huge hurdles, how did gene therapy even manage to recover? To find its footing again?
Well, the field took those critical lessons very seriously.
Yeah.
Researchers poured efforts into re-engineering the viral vectors to be much, much safer.
Okay.
And they also explored non-viral delivery methods, trying to minimize those immune reactions.
And the strategy changed too.
Fundamentally, the shift was from simply adding new genes to directly correcting the existing genetic errors within the body's own DNA.
Yeah.
And that intellectual shift really fueled the development of programmable nucleases, things like zinc finger nucleases, talons. They sort of set the stage
For CRISPR.
for CRISPR-Cas systems, yeah. CRISPR then just burst onto the scene. It proved to be far easier, more efficient, and crucially, incredibly more precise.
And that brings us right to CRISPR. I mean, most people have probably heard the name by now, but what exactly is this molecular marvel?
It's fascinating.
And how did we harness what sounds like something from nature into such a powerful tool?
Well, what's truly ingenious about CRISPR-Cas9 isn't just what it does, but where it came from.
Which is?
The ancient, really sophisticated adaptive immune system of bacteria and archaea.
Bacteria?
Seriously. Billions of years ago, these tiny microbes developed a defense mechanism to fight off viruses attacking them.
Okay.
They learned to identify the viral DNA, snip out little pieces, and then store those pieces, like spacers, within their own genome.
Like a genetic memory bank.
Exactly. It was their way of remembering past invaders, recognizing them if they came back.
So nature developed this brilliant find-and-cut defense system, and we basically just repurposed it. How did that happen?
Pretty much, yeah. Scientists like Francisco Mojica first described these clustered, regularly interspaced short palindromic repeats, that's CRISPR.
Right.
Then, a key breakthrough was identifying the Cas9 protein. It's a large enzyme with nucleus activity.
Meaning it cuts DNA.
It cuts DNA. And researchers realized Cas9 needed a specific tag called a protospacer adjacent motif, or PAM,
TAM? Okay.
right next to its target DNA to bind effectively. Crucially, they also found specific RNA molecules guided Cas9 to its target.
The CRISPR RNAs and the RNAs.
RNA, yes. The revolutionary simplification was combining these into one synthetic molecule. a single guide RNA, or sgRNA.
Making it simpler.
Much simpler. And that really opened the door to using CRISPR-Cas9 for editing genomes in, well, us, in mammalian cells. So think of it like this. It's an elegant two-part system. First, you have Cas9, the protein. Those are the molecular scissors.
Ready to cut.
Ready to make a precise double-stranded break in the DNA. Second, you've got the single guide RNA, the sgRNA.
GPS.
That's the GPS, exactly. It has a specific sequence, about 20 base pairs, that perfectly matches the target DNA right next to that essential PAM sequence.
You're guiding the scissors to the right spot.
To the exact genetic address you want to target. So once Cas9 makes that precise cut, the cell's own natural DNA repair machinery kicks in.
The cell tries to fix the break.
Right, and there are two main ways it does that. First is non-homologous end joining, NHEJ.
NHEJ. What's that?
That's the cell's quick and dirty repair. It's the most common pathway. It just sort of jams the broken ends back together.
Haphazardly.
Often, yeah. Which frequently leads to small insertions or deletions, indels, and that can disrupt the gene's function. Basically, it knocks the gene out.
Okay, so that's one way. What's the other?
The other is homology-directed repair, or HDR.
HDR.
This is the precise repair. It's less frequent, but way more accurate. The cell uses a similar undamaged stretch of DNA as a template to perfectly fix the break.
Like copying from a correct version.
Exactly. And for therapy, scientists can actually provide an extra engineered DNA template to the cell.
To make a specific change.
Yes, to encourage HDR to insert a desired specific edit right at the break site. That allows for incredibly precise genetic changes.
That whole process, it sounds incredibly powerful. A real leap forward.
Yeah.
But I imagine with that kind of power, the challenges, the responsibilities, they grow too, right?
Oh, absolutely.
What are the main hurdles researchers are still grappling with? And what does this mean for you, the listener, if you were considering this?
You're spot on. For this technology to be truly safe and effective in the clinic, there are critical limitations that need constant work. A major one is off-target effects, OTEs, meaning Cas9, as good as it is, can sometimes cut DNA at unintended locations, places that look similar to the target but aren't quite right.
And that could be dangerous.
It could lead to unforeseen consequences, yes.
So how are scientists making sure those molecular scissors don't, go rogue and cut the wrong thing?
Right.
Are they getting it to clinical safety levels?
They've been incredibly clever about enhancing precision. They've engineered different versions of Cas9, like Cas9 nickase. It only creates a single strand break, a nick. Use two of those, targeting opposite strands nearby, and you get much lower off-target activity.
Okay.
They've also developed high-fidelity Cas9 variants. These are much, much pickier about their target DNA. They only bind tightly if the guide RNA is a perfect match.
So better enzymes?
Yeah.
Anything else?
And optimizing the guide RNA itself, using computational tools to design guides that are less likely to match off-target sites. It all helps boost specificity. Another challenge is that protospacer adjacent motif, the PAM requirement we mentioned.
Right. Cas9 needs that specific tag next to the target.
Exactly. And the most common Cas9, SpeakCas9, recognizes a relatively common PAM, which is good, but the enzyme itself is quite large.
Making it hard to deliver.
Can make it tricky to package into things like AAV vectors, a common delivery vehicle. Other Cas9s are smaller, but they might have more restrictive PAMs, limiting where you can actually edit.
So it's like having a key, the sgRNA, but the specific lock, the PAM, isn't always where you need it to be. How are they broadening that target window?
Precisely. Great analogy. So scientists have engineered Cas9 variants to recognize a much wider range of PAMs, broadening specificity.
Giving them more locks they can open.
Basically, yes. Most remarkably, variants like SPRY have been engineered to be almost PAMless, theoretically allowing editing of almost any gene.
Wow.
Alternatively, there are RNA-targeting Cas variants, like Cas13. They target RNA, not DNA, so they bypass the DNA PAM requirement entirely, though that's usually for temporary effects, not permanent DNA changes. Then there's a significant safety concern, especially when making those double-stranded breaks. DNA damage toxicity.
The cut itself causes problems.
It can, yeah. Those breaks can trigger a cell's self-destruct program, apoptosis. And in certain human cells, like stem cells, this can even inadvertently select for cells that have shut down their p53 gene.
The tumor suppressor gene.
The very one. So that could potentially promote cancerous growth. Plus, sometimes you see unintended large deletions or complex DNA rearrangements near the cut site.
Yikes. Okay, that sounds serious. How are they making the process safer and more precise for actual clinical use?
This is where the innovation is truly incredible, moving beyond just simple cutting. Instead of full double-stranded breaks, some strategies use Cas9 nickase for single-stranded breaks,
Wait, mention that.
or even catalytically inactive Cas9, dCas9, for just temporarily turning genes on or off. But the real game changers for ultra-precision are base editors and prime editors.
Base editors.
Base editors are fascinating. They chemically change just one letter of the genetic code, say a C to a T or an A to a G, without making a double-stranded break at all.
Oh.
They fuse an inactive Cas9 or a nickase to a deaminase enzyme, which does the chemical conversion. It's revolutionary for single-letter fixes.
But limited.
Limited to specific base changes, yes, and they still have some potential off-target effects and delivery challenges. Still amazing, though. Then there are prime editors. This is really groundbreaking. It's described as a search and replace strategy for genome editing.
Search and replace.
Yeah. It uses a Cas9 nickase fused to a different enzyme, a reverse transcriptase, along with a specialized guide RNA. This lets it directly write in desired edits.
Like what kind edits?
Single base changes, small insertions, small deletions, all without causing a double-stranded break and without needing a separate DNA template.
So even more precise.
Unprecedented precision, potentially, for correcting specific mutations, even complex ones. And finally, briefly, immunotoxicity, our immune system.
Right, the body rejecting the CRISPR components.
Exactly. Many humans, maybe most of us, already have pre-existing antibodies against the common Cas9 proteins like SpeakCas9, just from encountering bacteria during our lives.
So our body might fight it off before it works.
It's a concern. And the viral vectors used for delivery can also trigger immune responses. So researchers are actively searching for Cas9s from less common bacteria, immune orthogonal variants that our immune systems might not recognize.
Okay, so we have this increasingly precise, powerful tool. But how do we actually get it into the body? Into the specific cells that need fixing? It's not like you can just swallow a CRISPR pill, right?
No, definitely not yet. Delivery is absolutely crucial, a major piece of the puzzle.
How do they package it?
Well, the CRISPR components, the Cas9 and the guide RNA, can be packaged in different forms, as plasmid DNA, as messenger RNA, or as preassembled protein RNA complexes, RNPs, ribonucleoprotein complexes. They're gaining traction because they act quickly and are cleared rapidly, which might help reduce off-target effects. It's a transient delivery.
And then getting those packages into the cells.
Well, viral vectors, especially AAVs, adeno-associated viruses, are still widely used. They're very efficient at getting into cells.
But they have those risks we discussed.
They do, including potential immune responses and size limitations. So non-viral methods are also used, like electroporation, using electrical pulses, or microinjection.
But they have downsides too.
Often, yeah. Electroporation can be toxic to cells, and microinjection is technically difficult, usually only feasible for lab settings or ex vivo work.
Okay, and crucially, where do these edits actually happen? Inside the patient's body or outside?
Right. There are two main strategies there. The first is ex vivo editing.
Ex vivo, outside the living organism.
Exactly. It means cells are taken out of the patient, genetically modified in a lab, checked, and then reintroduced into the patient.
What's the advantage of that?
Well, it offers potentially greater safety because the patient isn't directly exposed to the editing machinery, the virus, or whatever delivery method is used, and it allows for much tighter quality control before the cells go back in.
Makes sense. But what's the limitation?
It's really only feasible for cell types that you can actually take out, keep alive, expand in culture, and then successfully put back. Think hematopoietic stem cells, blood stem cells, or T cells for immunotherapy.
So not useful for, say, liver disease or muscle disease?
Generally not, or at least it's much more challenging for many solid tissue types. So that brings us to the second strategy, in vivo editing.
In vivo, inside the body.
Yes. This involves delivering the CRISPR components directly into the patient where the editing happens right there inside the body.
And this is necessary for?
It's essential for treating diseases affecting tissues you can't easily take out and put back. Muscles, the brain, the liver, the eye, many organs.
But that sounds harder.
It definitely comes with its own set of challenges. The therapeutic cargo might degrade before it gets there. The immune system might clear it out.
Physical barriers.
Getting past biological barriers like the walls of blood vessels to reach the target tissue, and then ensuring it gets distributed evenly within that tissue. It's a complex delivery problem.
What's really fascinating, though, is how quickly these developments are actually translating into human trials, into treatments.
Yeah.
Where is CRISPR making a tangible impact for patients? Like right now.
It's moving incredibly fast, you're right. CRISPR gene therapy is already making a difference in several severe conditions.
Such as
Well, for cancer immunotherapy, the first U.S. phase 1 trial using CRISPR-Cas9 ex vivo opened back in 2018.
editing T cells.
Exactly. Editing a patient's own T cells. They knock out the natural T cell receptors, knock out another protein called PD-1, which can act as a brake on the immune system,
Uh-huh.
and then they use a different virus, a lentivirus, to introduce a new gene that helps the T cells specifically target cancer cells. That's probably the most widely explored strategy right now in oncology. We're also seeing truly remarkable success in gene disruption for blood disorders.
Like sickle cell anemia, beta thalassemia.
Precisely. Companies like Vertex Pharmaceuticals and CRISPR Therapeutics have a therapy, CTX001, that uses ex vivo CRISPR-Cas9.
What does it do?
It disrupts a gene called BCL-ANA in the patient's hematopoietic stem cells. Disrupting that gene allows the body to start producing fetal hemoglobin again.
Which helps counteract the effects of the disease.
Exactly. It can provide significant clinical benefit for these major hemoglobin diseases. The early results from patients have been incredibly promising, really life-changing for some, and in a really major leap forward for in vivo gene therapy.
Editing directly inside the body.
Yes. There's a clinical trial underway for Leber congenital amaurosis, or LCA. It's a genetic eye disease that causes blindness in childhood.
And they're using CRISPR.
They're using a therapy called EDIT-101. It directly delivers CRISPR-Cas9 into the retina to correct a specific mutation in the CEP290 gene.
Wow. Offering hope for
Potentially offering a cure for genetic blindness, which
Yeah.
is just, a truly thrilling prospect.
This level of precision, this level of intervention, it inevitably raises huge questions, doesn't it? With a tool this powerful, they can literally rewrite our genetic code. Where do we draw the ethical boundaries?
The ethical discussions are incredibly complex, and a really key distinction needs to be made between somatic editing and human germline editing.
Okay, break that down. Somatic editing is
Somatic editing means making genetic changes only in the body cells of an individual, like liver cells or blood cells or eye cells. These changes are not passed on to their children. They're confined to that one person.
and that's generally considered.
With proper informed consent and for therapeutic purposes, treating a disease somatic editing is generally permitted and is where all the current clinical trials are focused.
Okay, then what's germline editing?
Human germline editing involves making changes to sperm, eggs, or embryos. These changes are heritable. They would be passed down through generations, affecting the entire gene pool, potentially.
And that's where the controversy really lies.
Immensely controversial. Because of the irreversible multi-generational effects and the fundamental removal of autonomy, of consent, for all those future individuals who would inherit the changes.
This immediately brings to mind the CRISPR babies case in China. That drew so much global attention and condemnation. Can you remind us what happened there?
Yes, that case, led by the researcher Jiang He, involved the genetic engineering of the CCR5 gene in human embryos.
With the stated goal of?
The intent was supposedly to confer resistance to HIV infection, but the ethical concerns were just profound.
Why specifically?
Well, first, the edits themselves weren't clean. They introduced random, uncharacterized mutations rather than precisely replicating a known, naturally occurring beneficial mutation in CCR5. It wasn't even clear if the resulting protein would function as intended or function at all.
Huge safety.
Huge safety unknowns, potential off-target effects cropping up later in the children's lives. Plus, one of the twins only had edits in one copy of the gene, yet both embryos were implanted.
And the rationale itself was questioned.
Deeply questioned. Using this powerful, risky technology for HIV prevention, something for which effective treatments and prevention methods already exist, rather than tackling a severe, untreatable genetic disease. It seemed irresponsible to many. That whole incident really starkly highlighted these profound questions about balancing scientific progress with responsible application.
Yeah.
There's a widely held fear, that if we normalize using CRISPR for maybe less debilitating conditions, it could act as a gateway, a slippery slope towards using it for non-medical purposes.
Like altering genes for aesthetic traits or enhancements.
Exactly, which reinforces this pressing need for really careful societal consideration. Who gets to decide CRISPR's therapeutic future, and where should those ethical lines ultimately be drawn? Big, big questions.
Okay, let's shift gears slightly. Beyond the direct therapeutic potential, it turns out CRISPR has found an unexpected role. It's actually contributing to the fight against pandemics. including COVID-19.
It is. It's a fantastic example of CRISPR's versatility, moving beyond just editing
That's
genes.
cool. In diagnostics.
Yes, primarily in diagnostics initially. A CRISPR-Cas12-based assay called SARS-CoV-2 detector was developed very rapidly
For COVID testing.
for COVID detection, yeah. It's designed to offer a pretty short turnaround time, around 40 minutes, and showed high accuracy in studies.
How does it work?
It basically targets specific viral RNA sequences unique to SARS-CoV-2. When it finds them, it triggers a reporter molecule to release a signal, often fluorescent, so you get a visual readout.
Okay, diagnostics. What about using it as an actual treatment for something like COVID?
Yes, that's also being explored, which is really exciting. Using the RNA targeting Cas13 enzyme.
Cas13 targets RNA, not DNA.
Right. So a system was proposed called Pac-Man. Stands for Prophylactic Antiviral CRISPR in Human Cells.
Pac-Man. Okay.
It uses a variant of Cas13, designed to simultaneously target and degrade multiple important regions of the viral RNA
Why multiple regions?
to make it harder for the virus to escape through mutation. This offers a much-needed potential pan-coronavirus strategy, which is vital given how quickly viruses like SARS-CoV-2 mutate.
So it's almost like CRISPR returning to its original job.
Battling viruses. Kind of, yeah. But this time, repurposed directly for human benefit.
Now, let's bring this conversation right to the cutting edge, to a development that truly feels like it shifts the paradigm. Something unfolding right now in 2025.
Yeah.
Tell us about baby KJ.
Yes, this is truly a historic medical breakthrough, a real testament to what personalized medicine can achieve.
What was the situation?
Infant KJ was diagnosed very shortly after birth with an incredibly severe, life-threatening genetic disease. It's called severe carbamoyl phosphate synthetase 1 deficiency, CPS1 deficiency.
And what does that do?
Basically, his liver can't properly break down the byproducts of protein metabolism. This leads to toxic ammonia building up rapidly in his body.
Which is dangerous.
Extremely dangerous. It can cause severe, irreversible brain and liver damage very quickly. It's typically fatal without a liver transplant.
And transplants are risky, especially waiting for one.
Incredibly high risk for these infants during that waiting period. So a dedicated research team at Children's Hospital of Philadelphia, Sheet Shop, and Penn Medicine did something extraordinary. They developed and safely delivered the world's first personalized CRISPR gene editing therapy specifically designed for KJ.
Personalized? How so?
They didn't just target the gene, they corrected his specific unique mutation within that gene in his liver cells.
Using what kind of CRISPR?
They used a base editing therapy. Remember, changing just one letter without the double-strand break.
Right, the super precise kind. And how did they deliver it?
Delivered via lipid nanoparticles, or LNPs. Those are tiny fat bubbles that can carry the therapy into the liver cells.
And the timeline, you said it was fast.
Astonishingly fast. The entire process from his diagnosis to designing the bespoke therapy, manufacturing it, and administering the first treatment took only 6 months.
6 months for a totally custom-made therapy.
Exactly. A bespoke CRISPR therapy, for one, because it was designed precisely for his disease-causing variant. This wasn't a therapy for a common disease, it was just for him.
That's incredible. Truly personalized medicine in action. So how has KJ responded? How's he doing?
The results reported so far are incredibly promising. He got his first dose in February 2025 when he was just 6 months old, and he's had follow-up doses since.
Wow.
He has tolerated increased protein in his diet, which is huge. He's needed less medication to keep his ammonia levels under control.
That's fantastic.
And crucially, really crucially, he has been able to recover from common childhood illnesses, like a simple cold, without experiencing that dangerous ammonia buildup.
Which normally would have been.
Extremely perilous for a child with his condition. It could trigger a life-threatening crisis.
So cautious optimism.
Very cautious optimism. Obviously, long-term follow-up is essential, but his doctors are encouraged. This marks the first known case of such a personalized, single-patient, CRISPR-based medicine being administered. And very importantly, it was designed to target only non-reproductive somatic cells, no germline changes.
Wow. So we've really covered a lot of ground. We've journeyed from those early challenging days of gene therapy
Definitely challenged.
to the incredible precision of CRISPR. We've explored its ingenious mechanisms, how scientists are overcoming limitations,
The off-target effects, the delivery hurdles.
and seeing its impact now, from cancer treatment and blood disorders to pandemic diagnostics.
And now, this breathtaking reality of truly personalized gene therapy, tailored for just one patient like baby KJ. It's here.
It really is.
This feels like the promise of gene therapy finally coming to fruition, doesn't it? It's truly a testament to decades of scientific collaboration, persistence, innovation,
Yeah.
and it's opening doors for patients with rare diseases, people who previously had maybe no treatment options at all.
So wrapping this up, what does this all mean for you, the listener? As this CRISPR technology continues its incredibly rapid advance,
It's moving so fast.
how will society grapple with making sure that such potentially life-changing, highly personalized therapies are actually accessible, equitably accessible to everyone who needs them?
Especially given, the likely complex pricing, the regulatory approvals.
Right. And as we gain even greater power, greater precision, how will we collectively decide where those ethical boundaries of human genome editing should ultimately be drawn?
Particularly as the capabilities become more precise and widespread.
It is. It's a lot to consider.
Yeah.
And definitely something for you to keep exploring on your own.